Okay, good afternoon, everyone. It's my great pleasure to introduce the keynote speaker for this evening. It'll be the last talk of this evening. So it's my great privilege to introduce Nadia El Mabrouk.
Perfect.
From the Department of Informatics and Operations Research at the University of Montreal. She's in informatics and operations research, but really all of her work has been in computational biology, focusing on developing algorithmic and mathematical models for computational biology. She's done quite a lot of impressive work in comparative genomics, including reconstruction of ancestral genomes, phylogenetic tree analysis of various forms, haplotype analysis, gene order, gene family evolution. I think these are going to become, they've been historically extremely important, and even more so now that we're seeing so many new genomes being sequenced every day. So I think these are going to be extremely relevant topics for the future, even beyond what they are today. She's been involved in many of the key conferences in the field. You may know her from all of her great work at RECOM and ISMB, WABI, so forth. So tonight she'll be talking about the future of algorithms for gene family evolution prediction, something I look very forward to.
So thank you very much for the presentation. Okay, so I will be talking about the evolution of gene families. So in the course of evolutions, genes evolve through microevolutionary events at the sequence level, but also at macroevolutionary events affecting the global the genome. Okay, so here I am interested in inferring the evolution of genes, accounting for various kinds of information. So this will be my content. So I will first begin by a motivation part, telling you why this field is important. And then in this presentation, I will focus on, okay, so my presentation is not, you will not have much probabilistic algorithms. It's more like John, it's more an optimization, you will more see optimization problems here. But anyway, here I am interested more by integrative methods for constructing gene trees, which are a method integrating the information in addition to the sequence information on the species tree, the evolution information. And this is done through reconciliation, by reconciling a gene tree with a species tree. So I will begin by reminding the reconciliation model. And then I will make an overview of existing methods for reconstructing, integrative method for constructing gene trees. And then I will talk about some challenges. For example, the existence of various sources of errors. We have to account for various sources of error at the different levels of sequence analysis. And we have also to account for various sources of information. So here I will more talk about the evolution information from the species tree, but we can also incorporate other information such as the gene order information, already known orthology relation between genes that we have to account for, or functional relation between genes. And then I conclude with other challenges. So genes are the building block of a cell during evolution, and of course it's important. They are the main target for understanding biological mechanisms. And during evolution, so they are mutated at the sequence level. So here it's an alignment of protein sequence. So we can see mutation at the sequence level. But they are also duplicated, lost, and also passed to organism through speciation or horizontal gene transfers. So we want to understand this evolution because most biological discoveries can only be made in light of evolution. And thus inferring gene relation is a prerequisite for functional prediction purposes. So for example, here is Manuel, my student Manuel. It's not, he put his picture of himself. I'm not responsible of that, of course. So the green letters here correspond to gene copies in gene families that play a role in eye color, for example. Okay, so we have 2 copies of the gene in the orangutan, 2 in the rat, 1 in human. So this is a gene families. These genes are homologs. They are assumed to come from to have evolved from a common ancestor, and it's to know. For example, we want to know, or if we study the rat, then we have to know that to which copy of the rat or to which copy of the orangutan the copy in the human. The human gene does it correspond to O or prime in the orangutan? So you notice that I'm not English speaking, so I'm French speaking, so you will have to afford my accent. Okay. I will do my best anyway. So it's important to know exactly which one is the corresponding copy in the orangutan. To know that, so the question that we ask is about the evolution of the genes. So this is, for example, a possible evolution. Okay, so let's say this is the common the ancestor in the supermammal. So this ancestor has been duplicated, and then we had 2 copies. So a rectangle corresponds to duplications and circles to speciation. So here we have an ancestral gene that have been duplicated. So the 2 copies have been passed to the 2 branches to the mammal branch and the rodent branch, and then this gene here was lost in this branch, and then we have a duplication in the rat branch here, etc. So this depicts a history of the genes. So if we remove lost branches and we make it nicer, so the tree representing the evolution of the genes look like this. So we see here that H and O prime, the least common ancestor of H and O prime, is a speciation node. And when homologous genes are orthologs, if their parental origin is a speciation. So these 2 genes are called orthologs. Because their least common ancestor is a speciation. They come from the same ancestral copy through a speciation, while H and O here, the least common ancestor of the 2 genes, is a duplication event. So these 2 are paralogs, and so they are 2 copies of 2 different, they are ancestor of 2 different copies of an ancestral gene copy. Okay, so here to answer the question that I have asked before, to which gene H correspond to? So H, the gene of the gene families in the human, correspond more to the copy O prime in the orangutan, because these 2 genes are orthologs. And so why is it important to know about the orthology-paralogy relation between genes? Mainly due to the orthology conjectures that says that at equal sequence divergence rates, orthologous genes are more likely to preserve the same function. So orthology is related to function, so that's why it's important to know about orthology. Okay, so inferring gene trees and gene relations is a prerequisite for many biological studies. So here is, for example, one project that I have been involved in. So it gives an example of why it's important to have accurate gene trees and to infer accurate orthology and paralogy and xenology relation between genes. So this is a project about the sequencing the nucleotide genome of Razzia stricta. So Razzia stricta is a plant growing in dry zones, such as in Saudi Arabia. And it produces alkaloids, which are chemicals that are used in chemotherapy protocols, and which is very difficult to replicate in laboratory. So the pathway producing these chemicals is difficult to replicate. So it's important to have new plants able to produce these chemicals. And it's also important to study this pathway to see what are the genes that are involved in these pathways. So it was important to have accurate gene trees and accurately labeled as duplication, as speciation and duplication nodes. So we have, there are 8 gene families that are genes that are known to be involved in this pathway. So each color corresponds to one gene family. So for each gene family, so we reconstructed the evolution of each gene families in related species. And for example, here it's important, so the red genes here are the copy in the Razzia stricta genomes. So for example, here we see that this gene is the one which is paralog to this gene in the coffee genomes. It's not paralog, ortholog. So this is an ortholog of this gene that is known to have a role to be implied. It's me. So, I don't know. Okay, so anyway, so it's important. So this one, for example, we found the one that corresponds to that in coffee. Okay, so this was my motivation. Okay, you see here we have labeled trees. So we have squares for duplication and circles for speciation. So we have, so this is obtained by reconciling a gene tree with a species tree. So gene pairwise relations between genes are a tree-based method for inferring pairwise or relation between genes are based on predicting first a gene tree according to an alignment of sequence based on using a sequence-based method, and then we use reconciliation. So given a species tree, if we know a species tree for the species of interest, then we can label nodes of the gene tree as duplication, speciation, or horizontal gene transfer nodes, and this gives us the relation between genes. Okay, so this step here is reconciliation. Okay, so reconciliation consists in embedding a gene tree in a species tree. So let's say we have our gene tree here, and this is the species tree. So the small letter here, so the leaf or G are genes, each belonging to the species. The small a here correspond to one copy of the gene family in genome A. Okay, so on the left they are genes, and on S letters correspond to species. So how to assume that the gene tree is correct, the species tree is correct, how to explain this gene tree. So to do that, we embed, let's embed the gene tree in the species tree. I just copied it in the species tree. So it gives this, so A, B here, is connected with C here, okay? And now how to explain that? I can assume that we had one duplication here, an ancestral gene has duplicated here, and I had another duplication here, and the gene have got lost here, etc. And this evolutionary history is consistent with this gene tree. And actually, this evolutionary history has a minimum number of duplication and loss events. It has 2 duplications and 6 losses. And this can be, and anyway, so this is easy to obtain from an algorithmic point of view. There is a linear time algorithm to obtain such a minimum history explaining the gene tree with a minimum and using a minimum number of duplication and losses. To do that, we use LCA mapping. So we have to label the nodes of the gene trees according to the nodes of the species tree. So a node of a gene tree is labeled by the least common ancestor of the leaves of the subtrees rooted at this node. So for example, here, this node is the root of the subtrees containing leaves A and B, and the least common ancestor of A and B is alpha, so I put alpha here. CAB, the least common ancestor of CAB here is gamma, so this is gamma, and this is gamma, and this is gamma. And then a duplication node, a node is a duplication node if and only if it's mapped to the same node than one of its children. So this node is a duplication node because it's labeled as these children here. This node is a duplication node because it has the same mapping as its children here. But this, that's all. And this is a speciation node. Okay. So this mapping is easy to do. And in linear time, we have a reconciliation minimizing duplication and losses. Yes, and it's not it's not difficult to add losses. Okay. So we can account. So here we just have, I just put the duplication and losses. We can also account for horizontal gene transfer events. So these are, so a horizontal gene transfer event is an event transferring one gene from one genome to another coexisting genome. So it's a horizontal movement, not a vertical one, between 2 coexisting genomes. So for example, here at that time, this genome has passed its gene to this genome here. And this evolutionary history here can be represented by this reconciliation tree here, where white circles represent horizontal gene transfer. So the reconciliation is harder with horizontal gene transfer because in the case of horizontal gene transfer, some evolutionary history are inconsistent, chronologically inconsistent. So for example here, this history here involves a cyclic inference, which is chronologically inconsistent. And this also, this evolutionary history is also inconsistent with the chronological, with the dating of the species tree. So in this case, we do not have cycle, but if we know if the species tree is dated this way, then we cannot have this horizontal gene transfer because these 2 genomes did not have coexisted. So in the case of horizontal gene transfer, so there are probabilistic models that are quite good, but also very slow. From the optimization part of view, for the most parsimonious reconciliation point of view, the problem is still polynomial time if we don't care about time consistency. But if we want to have also time consistency, then the problem becomes NP hard. Okay. And I will not talk about deep coalescence, but there are also the reconciliation model have also been studied in the case of the presence of incomplete lineage sorting of deep coalescence events. This is in the case where the gene is not completely fixed, when there are many alleles that are present in a population. And so these represent the evolution of a population of alleles. And so in this case, an apparent duplication may actually be just the fact that we chose different alleles in the population. So reconciliation in the presence of ILS will give less duplications than when we don't account for when we don't consider the possibility of deep coalescence events. Paralogs in different species can always be considered as coming as the result of an ILS event. But of course, if we have two paralogs in the same genome, then we should consider that there were an actual duplication, giving rise to a new locus of the gene. Anyway, so this problem, considering ILS, the reconciliation problem of minimizing is NP hard, even when only duplication events are minimized. Okay, so this was a review of the reconciliation concept. Okay. Yes, so I showed, until now, I showed you that reconciliation is useful to label the nodes of a gene tree, but we can also use reconciliation in the course of inferring a gene tree. And actually, it has been shown that including the species tree information during gene tree reconstruction can tremendously improve the accuracy of the gene tree. So I would talk, so for example, that's what, when we reconstructed the gene trees of the Razzia stricta genomes, we considered such an integrative approach, accounting for and we accounted for the information of the species tree. So actually, we had an ad hoc pipeline. So I can show you what we did. So we begin with a tree, with a FML tree, with a branch support with bootstrap values. So FML gives bootstrap values, statistical support for branches. From this statistical support, so this statistical support tell us which branches we are confident in and which one we are not really confident in. From this tree, we construct a population of trees by considering different routings and different contraction of branches using different thresholds. So a threshold, different threshold would give us different non-binary gene trees. And then using the species tree, we resolve this. Okay, so we use the reconciliation to resolve these non-binary gene trees, to make them binary in a way minimizing a reconciliation distance. Okay, so then from these trees, we keep only those that have good likelihood. So, but because we don't want to miss the closeness to the sequences, so we just keep those with a good likelihood and reconciliation scores. Anyway, and then we get a consensus tree from there, and we just take the tree which is closest to the consensus. So this was the pipeline that we used for this genome here. But in general, so there are many methods which I call integrative that act actually in two steps. So reconstructing one initial tree from the sequences and then refine the tree using the species tree information. And I sketched them on this picture here. So I will go through this picture here. So let's say, for example, we have methods here looking around, traversing the space of neighboring trees obtained from an input tree by performing branch swaps. Okay. So this is the case of Notung, tree fix, Muglien, and I. So from an initial tree, we consider all the trees that are at a given distance from the input tree by a given distance, considering some branch swapping. So the classical branch swapping are NNI, SPR, or TBR. So we explore the space of neighboring gene trees using heuristics such as branch and bound or hill climbing. And in the worst case, we can end up exploring the complete exponential size tree space. So this is one class of algorithms in the integrative methods. Another way of doing is from the input tree. So this is more what we did in the lab, and this is more the method on which we constructed the way we constructed our trees in the Razzia stricta genome. So from this we contract weakly supported branches, and then we obtain a non-binary tree, and then we resolve this tree in a way minimizing a reconciliation distance. Profile NG is our algorithm, and Notung is the algorithm of Dany Durand, from here. So for example, from this tree here, if we take the threshold of 90, so we will contract these branches here. And then we want to resolve this polytomy, actually the tree, in a way minimizing a reconciliation distance with S here. So we can see if we have no horizontal gene transfer. With horizontal gene transfer, it doesn't work, but with no horizontal gene transfer, we can proceed independently with each polytomy. So we will resolve this polytomy here, and then this one, and then this one. So if we proceed this way, it's an optimal way of doing. Okay. So we have a polytomy solver. It's a linear time algorithm that can resolve a non-binary tree, minimizing a duplication loss reconciliation. Okay, and resolving a non-binary. Yes, so we are actually here, we are considering the problem of reconciling a non-binary gene tree with a binary species tree. So the problem is linear time for duplication and loss, and is NP hard with horizontal gene transfers. But there are heuristics that are implemented in Notung in particular. Okay. Okay, we have another class of integrative methods using amalgamation. Instead of keeping just one input tree, we can keep a family of trees, for example, equally likely trees that can be output from a Bayesian method, for example. And from this tree, we will we amalgamate a tree. So an amalgamated tree is a tree in which each cluster has to belong to one of the input tree. And we do that in a way minimizing a reconciliation distance. This is Céline Skornavaca in French works a lot on this type of methods. And finally, so here the trees here, the input trees, are on the same data set. So we have the gene families and many different trees for this gene family. But we can also cut the gene family into different orthologous groups, for example, and construct separate gene family for subset of the gene families that can be overlapping actually. And obtain a set of gene trees, and then the problem is to construct a super tree, so a tree containing them all, and minimizing the reconciliation distance. So we worked on this type of problem in our lab. Okay, and this here, so this problem is interesting if we have different trees for different subset of the gene families, but we can also use it, for example, if we have an input gene tree and where the upper branches are not very accurate. Okay, so here, for example, if we remove these branches, we obtain a set of gene trees, and then the problem is to find a tree. This a super tree, so a tree containing all these subtrees, which agrees with the species tree. Okay. So finding a super tree containing a set of input tree. So to do that, actually, to have a tree containing all the subset of trees, we can just do that if it's not always possible. The trees has to be consistent. And we showed that the problem, so it's NP. The problem is NP hard for the duplication loss reconciliation. So given a set of consistent subtrees, finding the super tree minimizing the duplication loss distance is an NP hard problem, but we have algorithms to do that. Okay, so this was a summary of the integrative, the different frameworks for the integrative method for constructing gene trees. So what we can do, although we have good methods, many things remains to do. For example, we have the problem of various sources of errors. So we need, before reaching the trees, we need to get the sequences, annotate the sequences, align them. So there are many treatments that we have to do on the data, and each step involves, we have errors at each step. And actually, well, here I show about reconciliation. Reconciliation, it's a good method if the trees, if we trust on the gene tree and on the species tree. Otherwise, we can't get, if the gene tree is not accurate, then the evolutionary history that we get with the reconciliation can be completely out of the point. Okay. So, for example, here, even if I don't take the right routing. So, for example, here I have this gene tree here. If I take this routing, then the tree will be this one, and reconciliation will give me one duplication at the top. And if I route it here, so I will have this, I will get this tree, and now the duplication is here. So it's a different tree, and it's a different history. And also it gives different relations, pairwise relation between genes. Here A and C2 are paralogs, while here they are inferred to be orthologs. So just misrouting the tree can give wrong results. Okay, and in general, so precision depends on the sequencing step. So the good point is that we have better methods for sequencing, and and with the high throughput sequences we have less missing data, so that's good. But we can have errors in the sequencing step. We can have errors in the annotation step due to errors in the homology search tools, and even in the genetic code evolution, because so when we translate a nucleotide sequence into an amino acid sequence, we use a genetic code. And if we don't choose the right genetic code, so the sequences can be completely different. Okay, and I will actually talk more about this fact here, because checking codon reassignments is not a routine. It's not done in the pipeline for the sequence annotation, and I will show that it can give many wrong predictions. And we can have errors in the gene clustering step also when we reconstruct gene families. Okay, and and precision depends also on the alignment step and on the phylogenetic reconstruction step. Okay, let's talk about codon reassignment. Here is a tree of metazoan mitochondrial DNA. And all the colored squares here correspond to codon reassignments. So for example here, ATA is reassigned from in this genome here is reassigned from isoleucine to methionine. Okay, so these are all reassignments, codon reassignments. So if we don't take we don't account for these codon reassignments, we end up with completely wrong phylogenies. So, for example, this is the tree. This shows the topological difference between metazoan phylogenies inferred from 13 mitochondrial protein. When we use the mitochondrial default code that is given in database, versus the code when we account for all these codon reassignments that have been reported in the literature. Okay, so we see that the difference is significant between the two phylogenies here. So it's important. So inferring predicting codon reassignment is something that it's important to do. And in our lab, we have a program called Core Tracker for predicting codon reassignments, which is a non-parametric random forest, an algorithm based on random forest, which detects unusual and recurrent amino acid substitution, detect codon reassignment from an alignment, from a protein alignment. From the conserved position, in the conserved position of a protein alignment. Okay, and Core Tracker have different validation step. So when we infer one prediction, we use the new code and translate and you see if the new alignment is better than the old one, etc. Okay, but what is important to do also is to check with the tRNA, because the tRNA has a major role in translating one codon into the amino acid. So if there is a codon reassignment, there should happen something with the tRNA corresponding to this codon. So it's important to look at the tRNA. So for example here, this is the standard structure of a tRNA tryptophan. But in, I don't know which species here, this tRNA tryptophan is clustered with tRNA cysteine. And when we look at the order of genes on the genome, actually it looks like this tRNA cysteine is a tandem duplication of tRNA tryptophan. So actually this one here, the TCA, does not correspond to tryptophan, but to cysteine here. Okay, so we have validated, so in this case, using the tRNA phylogeny and the gene order, we could validate this codon reassignment, and we can also infer the tRNA evolution that gave rise to this evolution. Okay, so actually this is not automated. It's very difficult to make this step automatic for many reasons. First, it's not always possible to have a nice phylogeny of tRNAs because there are few positions in the tRNAs that are a few different positions in the tRNA. Anyway, so it's not always easy to find a good phylogeny of tRNAs. And anyway, this step is not automated, so it remains to automate this step of checking with the tRNA phylogeny. And this step of looking at the order of tRNA is not automated either, because actually we don't have such an algorithm for inferring the evolution of tRNA repertoire evolution. So an open problem in this field would be to infer a method, to develop methods for inferring tRNA evolution under duplication, loss, rearrangement, but also a tRNA shift of identity. Okay, so this is a small parsimony problem, which has not been really addressed because usually this remodeling part is not considered in classical small phylogeny problems. Okay, so you wanted challenges, so I tried to do my best to find open problems. Okay. So this, yes, I agree that I want a little bit far from the tRNA from gene tree reconstruction, but if we have the wrong genetic code, then anyway we will have the wrong gene tree, you know. The second problem is, which is interesting here also. Okay, so we infer codon reassignment when we see that there is a difference between the nucleotide sequence and the inferred amino acid sequence. Okay, so but the difference between a nucleotide sequence and the corresponding amino acid sequence can come not only from codon reassignment, but it can due to different events. It can be due to post-transcriptional modifications. So the messenger RNA can be affected by such modifications. So some codon reassignment actually are not codon reassignment because they are actually mutated in the mRNA itself. So if we have the mRNA sequence, we can see that there is no difference, that it's okay, it's not a codon reassignment. But if we don't have the mRNA sequence, what to do to make the difference between a codon reassignment, a substitution, an mRNA editing event? So this is really an interesting problem that has not been studied much right now. So, for example, here I have, this is a set of algal species, I think. And here, this reassignment, this is the result of core tracker. But all these predicted reassignments, are codon reassignments, are actually from arginine to tryptophan, are not codon reassignment. Actually, they are caused by an RNA editing in plant mitochondrial DNA from C to U. So here, CGG doesn't stay like that in RNA, in messenger RNA. The C becomes a U in the messenger RNA. So there are many examples of that. So how can we make the difference between codon reassignment and RNA editing? But one thing is that RNA codon reassignment is expected to affect the whole genome, because if the code, but editing just affects some positions in some genes, so we do not expect to see them spread in the genome. But still few is known about the property of RNA editing. So anyway, in the lab we are working on that to find an optimization of different ways to predict RNA editing. So we have a kind of pipeline that coming from core tracker. So if we have suspect cases of codon reassignment which are not validated by a core tracker, then we can see if they are compatible with some editing. We can check if they can work. Anyway, we are still at the beginning of this analysis here. And we can also add another challenge is to account for other type of data in the reconstruction of a gene tree, not only at the sequence level and evolution. We can also consider gene order, some known orthology relations, or a gene function. So here I will just give an idea about how we can integrate gene order in the inference of gene relations. If we have, for example, two centenic blocks, so these are two segments in two different chromosomes, two different genomes containing the same genes. And let's say we predict that, so if we have these two segments with the same genes, we can expect that they have evolved from an ancestral segment, an ancestral centeny. So either from a speciation event or from a duplication event. So we expect all of these, the pairwise relation between genes. We expect all being orthologs or all being paralogs. But let's say we have inferred with our reconciliation methods that all these pairwise here genes are orthologs, but here we have inferred a duplication at the least common ancestor of these two genes. Then we can say that this is a wrong paralogy because it's alone because if all the others are speciation, then it's more likely than the two segments have evolved anyway, are coming from a speciation event. So considering with such consideration, we in 2013 we have studied this problem, and we have noticed that ensemble gene trees, for example, contain many wrong paralogies and wrong orthologies. Around 31% of the trees contain at least one error of this type. And in 2013, we've got a polynomial time algorithm for correcting wrong orthology paralogy relations. Okay, so also an open problem. So reconciliation assume independent evolution of each gene families, but as we have seen here, segments, genes that are next to each other are more likely to have evolved together. So for example, let's consider this hypothetical evolutionary history here. So we have an ancestral segment of 3 genes that have been duplicated. So we have this, so that are duplicated in, okay, just, so this part here is duplicated. Okay, so in the genome, we have a duplication of these 2 genes somewhere else in the genome. And then, so the corresponding genome has these 2 copies of segments. And then after speciation, they are inherited in the 2 branches. And then we have another duplication, a full duplication here. Voila, and this means and losses. And we end up with this segment here. Okay, so this is again the history that I have shown you. So if we take the red genes, the corresponding gene tree is this one, A1, no, B1, B1 with B2, with A2, with A1. So this is the history of the tree that we get from the red gene family. And if we reconcile it with the species tree AB, we get this labeling here. And if we embed it in the species tree, this gives us this embedding here. And then we do the same with the 2 other gene families. And reconciliation, if we do just reconciliation as we okay, if we assume that each gene family is independent, then we get this forest of gene trees, and this doesn't reflect the history of the centenies from a single centeny. Okay, so we have to do better than that. So we have to extend the reconciliation model to segmental. So here, if we consider, it remains to consider horizontal gene transfers, but for duplication and loss, so we need a reconciliation model to extend the reconciliation model to account for segmental duplication and losses. And this is something that has not been done before. So we have a paper at RICOM CG in 2 weeks in Sherbrooke, in Quebec. In 2 weeks in Sherbrooke. Okay, about this extension here, considering segmental gene, segmental gain and loss events. So as an input, we have gene families grouped into centenies and 1 tree for each gene family. And we want to output a most parsimonious super reconciliation, which extend a super tree of G. Okay. Of course, a super tree does not always exist. So to have a super tree, the gene tree should be consistent. Otherwise, we have to do we have to make a modification anyway. So it's another problem. If the gene trees are not consistent, we have to do something else. But let's say the gene trees are consistent, then we have to get all the possible super trees for these gene trees. And then we show that we can label the species tree according to the labeling of the gene trees here. And then given the orders at the leaves, we have to consider all possible orders, gene order at the root. And then the main problem is actually, given such a tree, to label, it's a small parsimony problem, it's to label internal nodes and and find lost nodes also, minimizing the reconciliation cost. So we have to end up here, for example, with a search from this to this, and just this problem is, I don't know, the complexity is open actually for this problem. Okay. So there are many problems that remain to be explored here. For example, so duplication loss history is not always possible because here I didn't take into account rearrangements, and that's bad. When we look at gene order, we should allow for rearrangements. So right now we integrated some consideration of rearrangement, but still many things have to be done here. So if the individual gene trees are not consistent, we can't do anything with my method. If the gene order are not consistent, this method doesn't work, so it has to be adapted to account for rearrangements. The complexity of the problem is still unknown. So we have to account for rearrangement, account for tandem duplication too. So here I account just for transposed duplications. Because it's just an extension of the reconciliation, the classical reconciliation model. Okay, so the tandem duplication will add a segment inside here. Okay, with reconciliation, when I have a duplication, I create another segment. A tandem duplication would include something in one node anyway. It will not make a nice generalization of the classical reconciliation. We have to look for other anyway. So finally, I don't have much time. So we have various. Okay, so I have presented a lot of most parsimonious methods. Of course, probabilistic methods will be based on more realistic models will be nice, but it's not always easy to find the probability to give to each event. And the more events, the more complex it takes anyway. What is good with parsimony-based methods, except that they are simplistic hypotheses, but we can extend them more easily. Okay, with the parsimony method, we can add, they are more flexible to different data sets, to rooted and rooted tree, dated and dated tree, and they are easily extendable to different input information. So we can add gene order, we can add gene function, but the problem is to know how to integrate all these sorts of information in a single robust framework. In this presentation, I mostly showed you a two-step method. Okay, first predicting a gene tree from sequences, and then correcting the gene tree considering the evolutionary information. But ideally, we should have all together in one step. But one-step methods exist actually. So, for example, field dog here computes the joint likelihood associated with a substitution model and duplication transfer loss event rates. So the drawback is that they are computationally costly, and also they are subject to be stuck in a local minimum. So in the lab we have developed another algorithm based on genetic algorithm, which consider a multiple objective optimization based on the multiple objective optimization algorithm. And as we are considering a genetic algorithm consider a population of gene trees. So we are less neighborhood exploration of a population of trees decreases the risk of being stuck in a local optimum. So, but I don't know, I will have time to show you this algorithm. It's based on a genetic algorithm. So we have to show. And the fitness score is based on both sequence likelihood and a reconciliation score. We end up with Pareto sets. So at each step we have a Pareto set of gene trees that are not dominated by others. Okay, and for crossing and mutation, okay. So at each at each step, we have to do genetic. Okay, if I don't have time to explain, I don't have time to explain, right? So let's skip this part. So anyway, we have a nice algorithm. So GATC is here. It outperforms other methods. All these other methods here, these all are based on our integrative method, but based on a two-step. The two-step method that I showed you. All these methods are contained in my first the diagram of methods, and RAXML is it's a maximum likelihood reconstruction method. Okay, so other challenge would be to include other possible information, such as gene order, preliminary information, or orthologs. Also, how to combine both speed and accuracy, because as we increase accuracy, we increase speed also. So GATC ends up being slower, of course, than the other methods. Maybe parallel computing. Okay, so I still have many other open problems that I can't tell you about. Okay, and also one of the big problems also is to know how to evaluate reconstructing methods, because we always we don't know the reality of the evolution of the gene families. So we have to have simulated. We stand on simulations. So we are always biased by the model that we have used to make the simulations. So we need to have true trees, but there are not so many true trees. There are some gold standard trees, but there are not so much. In Swiss tree, there are just 19 trees and most of them are non-binary, moreover. So maybe we have to develop our own database of gold standard tree with all the methods that we have. Maybe it's time to have our own database of gold standard trees. And also how to compare trees, which distance to use. And here I am comparing labeled trees, trees labeled as and how to compare labeled trees. We don't have measures to compare labeled trees. And that's all. I don't have any conclusion. But I took the picture that I had from my lab. My students are not all here. Ida is my colleague. I work with her on different problems. She's organizing next Recom CG in 2 weeks in Sherbrooke. Here is Emmanuel, my PhD student. He's defending his PhD soon. And Manuel, whom many of you should know, he's now a colleague of Ida in Sherbrooke. Voilà.
So thank you very much for that. Let me take the privilege as moderator to ask the first question.
Yeah.
So I think it was step 4, you mentioned some of the sources of error.
Yeah.
And this is really forefront to me because our lab does a lot of de novo assemblies, and we do see with some of the newer technologies, there can be, 1% error or a fraction of a percent error. And I'm wondering if you've done any sort of evaluation on, how much error can be tolerated at the sequence level before it really starts to perturb, the inferred gene trees?
No, I didn't do such. I can't tell you. No.
So,
so I'm yeah.
so the inputs to algorithms, you start from the annotated genes, or do you start from the sequence?
It's much more a philosophical point of view.
Yeah.
Myself, I don't know. It depends on which problem I treat. Okay, my part of the study, it's more about on reconciliation. When I already have the tree, so it's more knowing which kind of error. But you see, when you have very small, the problem with reconciliation is that really it really depends on the tree that you give it. So even as I showed, giving a wrong routing will give a very different history. So yes, this is a big problem.
Okay, thank you. Other questions? In the back here?
Yeah, so just to follow up on that issue, like say I work on a set of non-model organisms and I have no access to a species tree. Right? It's just not known, right? I mean, it seems like you touched on that on the last question, but then I have sequence. I've sequenced this thing and I've used a fair amount and I've done de novo analysis and I've done some gene calling, and therefore I can do some sequence-based gene family creation, right? So I can get to the gene family step and I can potentially construct gene family trees, but I can't use any of the reconciliation techniques that you mentioned. But I am still interested in orthology detection. So is something like that possible, and if so, how would you do it?
But if you don't have the species tree, we can't talk about reconciliation, because reconciliation is really taking account, really embedding, trying to explain the observed gene tree given the species tree. But we can you can infer orthology from other information, from the gene order information, from the gene function. And then we have the problem if Emmanuel actually worked on the converse problem. If given a set of orthology relation that you have inferred from different data set, are they consistent? Do they correspond to a gene tree? Because you can't. This is also an interesting computational problem, a theoretical problem. Yes, you can probably infer orthology relation. Yeah, but there is a confusion also when we talk about orthology relation. Do we talk about a function similarity, or is here orthology is taken in the sense of the Fitch definition? It's based on evolution, but actually, some clustering, many softwares are clustering genes according to sequence identity, and the clustering based on sequence identity, they call them orthologs. So yeah, it depends on.
Resort then to sequence-based similarity or distance matrices as opposed to
yes. Here
orthology, right?
Yes. Here I'm talking about tree-based inference methods. Okay. If I don't have the species tree, I can't do anything that I showed here. The reconciliation just work with if we have the tree. But we can maybe we can have a non-binary tree. Yes, there are also. Danny Durand have studied the reconciliation with a non-binary tree, account with ILS. She considered ILS. Okay, so we don't need the species tree maybe to be completely resolved. But if you don't have any species tree, so this is not for you.
Any last quick questions?
So can I ask two or only one?
Maybe one, and then we can do the other one ourselves.
So the main assumption that you have here is genes are the basic units of unfairness.
Yeah.
But for example, I work with the SIG2, the 6 gene, And there's a lot of information exchange with some of the parallels. And in fact, some of the non-functional pseudogenes that are very similar. And obviously, the unit of information exchange is not the full gene. So when you, for example, look at...
It's not so domain, yes, yes. So extension on domain should, yes, it's important, would be important
I'm
too.
wondering if you have done the same thing you did in performing reconciliation based on the species tree on individual, say, exons based on the gene tree.
On exons, yes,
Or, yeah.
yes.
Have
But
you-
actually, the theory, yes, the computational problem, this will not make any difference for the computational
Yeah, of course.
problem.
I'm just
Yeah, for the
wondering, it's a
computational
biology.
problem, I don't, yes, it can, but from biology, from what this will give you, it depends on the data. We have to look at each data separately, actually. We can't assume, I can't tell you that we are selling one method for everything anyway. When they asked us to reconstruct this gene tree for the Razzia stricta genomes, we had to consider specifically these data sets. So we didn't just use an existing pipelines, you know. We have to adapt it to any data set. So maybe, probably, we will be able to get
There are follow-up
something.
questions, but we can take it offline. Thank you.
Thank you. All right, thanks very much, Nadia. And thanks to all the speakers today. Carl has a few comments.